I am here to represent Orchard Therapeutics and share a brief overview of our pipeline and proposed mechanism of action for all of our potential therapies. The vision at Orchard is to end the devastation caused by genetic and other severe diseases. We aim to do this by discovering, developing, and commercializing new treatments that tap into the curative potential of hematopoietic stem cell gene therapy. We're a global gene therapy company born out of academic partnerships between some of the world's most preeminent researchers and institutions in the area of ex vivo autologous gene therapy. Next slide. So here you see the Orchard pipeline as it now exists. If you had seen a version prior to 6 months ago, it would have looked a little different. In March, Orchard did announce a reorganization of our portfolio and a reduction in workforce, as did many other companies, to focus on hematopoietic stem cell gene therapy platform exclusively in severe neurometabolic diseases and early research programs. Moving forward, Orchard will continue its investment in adadarsigine autotemcel, say that 3 times fast, or OTL-200 for metachromatic leukodystrophy. We want to help support regulatory and future commercial activities for potential US approval and launch. We anticipate filing the BLA for OTL-200 the end of this year or early next year. We also will continue to advance clinical development for OTL-203, which is MPS 1H, and for OTL-201, which is for MPS 3. They kind of got those numbers mixed up a bit, but I have been able to keep track by knowing that they're mixed up. Both of the MPS studies are fully enrolled in proof of concept and ongoing. We also, as you can see, have some very early research programs that we won't be addressing today. Next slide. So here you'll see the figure for our proposed mechanism of action in our ex vivo genetically modified autologous CD34 positive HSC gene therapies. All of our programs utilize the same proposed mechanism of action. Just to get ahead a little bit, there were some questions earlier about hospitalization and timelines and transplant and chemotherapy. So I'll start with that before moving on through this particular figure. Ex vivo indicates that this requires for the cells to be removed by apheresis, then transduced, and then returned to the patient. Apheresis takes a few days. The transportation and manufacturing and quality control for the cells takes a period of 4 to 8 weeks. And during that 4 to 8 week period of time, the child does not need to remain in the hospital. They do return to the hospital once the cells are returned to a particular treatment center that has been qualified to deliver the therapy. That treatment center will administer a myeloablative conditioning regimen, typically busulfan, to the patient, and then administer the cells that have been gene corrected back to the patient. The patient then is required to stay in the hospital just as they would for an autologous bone marrow transplant for a period of time while they engraft and recover from the transplant, and then are followed as outpatients thereafter to monitor for any other signs and symptoms that might be concerning. So then on to the proposed mechanism of action. So autologous stem cells are collected, as I mentioned, by apheresis, the mobilized peripheral blood, and transduced with a lentiviral vector, which inserts 1 or more copies of the human ARSA with respect to MLD, complementary DNA, into the cell's genome. That genetically modified cells become capable of expressing the functional ARSA enzyme. When this is administered to the patient following the administration of a myeloablative conditioning regimen, and it is important that it's myeloablative because we understand that we need to make space both in the bone marrow and the CNS for these cells. So these cells are intended to engraft and then repopulate the hematopoietic compartment. A subpopulation of the infused stem cells and or their myeloid progeny is able to migrate across the blood-brain barrier to the brain and engraft as central nervous system resident microglia and perivascular CNS macrophages, as well as endoneural macrophages in the peripheral nervous system. Lentiviral mediated overexpression of the functional enzyme in these macrophages or microglia may serve to enhance the corrective potential of the damage response pathway. So the myeloid progenitors derived from the modified stem cells can cross the blood-brain barrier. They engraft and then they differentiate into these microglia cells that then produces the functional ARSA protein. And that functional ARSA protein that's been overexpressed can then be taken up by the oligodendrocytes. Gene-corrected microglia cells can address also the inflammatory and apoptotic components of the pathological microglial activation and reconstitute the scavenging microglial function of extracellular sulfatides, which are key elements in the pathogenesis of MLD. Following the successful and stable engraftment of the patient, remember they're hospitalized for that period of time after their transplant, the effects of the product are expected to be persistent. So this is a one and done therapy. They receive the transplant, they engraft, they recover, and they have persistent expression of the enzyme. Today, Orchard is advancing a pipeline spanning preclinical, clinical, and commercial stage stem cell gene therapies designed to address serious diseases where the burden is immense and current treatment options are extremely limited or non-existent. I'd like to thank all the patients that have dedicated their time and efforts and their families that have contributed to advancing stem cell gene therapy for patients. Thank you for your time. I'm happy to answer questions and hopefully I got ahead of a few of them.